The other thing, I guess the very traditional view of genetics is that our DNA has a very high determination on what happens. You know, if you have the particular genes for a condition, you're going to get that condition. That's how we tend to think of it classically. But we know more and more that there are some conditions which have a huge variety of genetic influence. So much more than one gene, say. If you have a condition like Huntington's disorder, that's one gene. And if you have the gene, you're going to develop that condition. But most things, is it fair to say that most things aren't like Huntington's?
It's very fair to say it. So some traits are exactly like Huntington's, like cystic fibrosis, or variants in the BRCA1 gene for breast cancer, where those genetic changes on their own put someone at a very high risk of developing a genetic disorder. And that's the sort of genetics that we're taught in school, and it leads us to think about genetics as a black and white, yes, no, we're at risk or we're not at risk.
You have the fat gene.
Exactly.
I have the fat gene.
Well, and can I say, we all have the fat gene. We all carry that gene. But what differs between us is the changes in that gene. So specific changes at the level of the DNA variants that we carry. And that's what makes a difference between us.
Tell me more about that.
So our genes are made up of a long string of DNA variants. So the base pairs A, C, G, and T. And about 99% of our genome is exactly the same. You, me, and everyone listening to this podcast will have exactly the same base pair at exactly the same place in our DNA. But 1% of our genome may differ between us. And our genome is over 3 billion base pairs long. So 1% still gives us quite a lot of opportunities to differ. And some of those changes have a very big impact on our risk of different diseases, different conditions. But most of those changes that we see in our DNA have a very modest effect on disease risk. And much of my research is in the genetics of depression. And for the last 10 years, we've been carrying out studies to identify the genetic contributions to depression. And we do this in a classic epidemiological design that's a case control study. We collect, we perform research studies with groups of people who've been diagnosed with depression. And we compare their DNA to people either from the general population or people that we know have not been diagnosed with depression to date. And we step along the genome, looking at multiple different places on the genome, and we look at the frequency of that change. And we say, okay, this site in the genome, people either have a C allele or an A allele there. What's the frequency of that A allele in people with depression and with people without depression? And then we do a very simple statistical test and say, does that differ? And we do that at several million places through the genome. And by carrying out really careful statistical tests, we can identify specific changes that seem to increase people's risk of developing depression.
So very specific genetic differences that are more correlated with someone having depression. Yeah.
Exactly. But the actual risk conferred by each of those individual changes is very, very low.
Yes.
We're not in the single gene Mendelian disorder territory that we've been with Huntington's disease. We're now in a polygenic genetic model, which is what seems to be underpinning depression, schizophrenia, bipolar, and also most physical disorders that really have an impact on people's lives. We're not looking for single genetic changes. We're looking for genetic changes spread across the genome, each of which have a very modest effect on people's risk of developing a disorder. But when we put them together across the genome, that gives us much more information on risk.